In this video, what I want to do is a quick summary of glycolysis, link reaction, and Krebs cycle. I hope it is not too overwhelming. So let's start off with glycolysis, which happens in the cytoplasm. First, the glucose molecule has to be phosphorylated with 2 ATP. What does it mean by phosphorylated? It means that you have to add some phosphate groups to the glucose. The reason was because the glucose is too stable. So by adding the phosphate from the ATP molecule, the glucose becomes something known as fructose 1,6-bisphosphate, and it's more reactive. And you had to spend 2 ATP. The fructose bisphosphate undergoes lysis, which is breaking down, and it becomes 2 triose phosphate molecules, and the triose phosphates will undergo substrate link reaction to produce 4 ATP, and it also undergoes oxidation or dehydrogenation, where it releases the hydrogen is accepted by NAD, and the NAD becomes 2 reduced NADs. And the end product of glycolysis is pyruvate. The pyruvate will then enter the link reaction in the mitochondrial matrix. That is the location where link reaction occurs. And the pyruvate undergoes decarboxylation, and it releases carbon dioxide. The pyruvate becomes a 2-carbon molecule. The 2-carbon molecule undergoes oxidation or dehydrogenation, where the hydrogen is accepted by NAD, becomes 2 reduced NADs, and then it forms acetyl group. The acetyl group will be added with coenzyme A because the coenzyme A helps to transport the acetyl group into the Krebs cycle. And the end product of link reaction is the acetyl coenzyme A, or acetyl-CoA. The acetyl-CoA will then enter the Krebs cycle, where the acetyl group, the 2-carbon molecule, will be bonded together with oxaloacetate, which is a 4-carbon molecule. By the way, this happens in the mitochondrial matrix as well. So when the acetyl group binds together with the oxaloacetate, it forms a 6-carbon molecule. And the name of the 6-carbon molecule, I told you before, you need to remember this. And the name of the 6-carbon molecule is called citrate. But what happens to the coenzyme A? The coenzyme A comes out. The coenzyme A is like, I've done my job, so I'm just going to transport other acetyl groups in the future. So the citrate molecule in the Krebs cycle, 6-carbon, undergoes decarboxylation, which produces carbon dioxide, and it also undergoes oxidation or dehydrogenation, which produces 2 reduced NAD. And the citrate, which was a 6-carbon molecule, becomes a 5-carbon molecule because each of them released 1 carbon, right? So it becomes a 5-carbon molecule. You don't need to know the name of the 5-carbon molecule. That 5-carbon molecule undergoes decarboxylation, dehydrogenation again, 2 carbon dioxide, 2 reduced NAD are produced. You have these 4 carbon molecules. Those 4 carbon molecules will undergo substrate link reaction to produce 2 ATPs, and they undergo dehydrogenation or oxidation again. And thus you produce 2 reduced FADs and 2 reduced NADs, and the oxaloacetate is regenerated. That is a very important aspect of the Krebs cycle. You spend the oxaloacetate, but you must regain the oxaloacetate no matter what. All right? So now the point of the matter is, what is this all about? This is us breaking down the glucose molecule. Because remember, the glucose molecule, as I've highlighted, was a 6-carbon molecule, right? So in that case, that 6-carbon molecule has been completely broken down. Don't believe me? Because look here. As you can see here, it produces the 2 carbon dioxide over there, it produces the 2 carbon dioxide over there, and the 2 carbon dioxide over there. How many molecules of carbon dioxide are produced? 6. Those 6 carbon dioxide came from the glucose. It has completely been broken down. But remember, we don't just want to break down the carbon-carbon bonds, we also want the hydrogen. Why do we want the hydrogen? Because we want the hydrogen in chemiosmosis. This process, chemiosmosis, to power the ATP synthase later. That hydrogen has been accepted by those reduced NADs and reduced FADs. And by the way, in the process of breaking down the glucose molecule, you also released some energy which gave you a little bit of ATP. Not much, but a little bit. It's okay. So in total, right now, from the breakdown of the glucose molecule completely through glycolysis, link reaction, and Krebs cycle, you have produced 6 ATP molecules, 10 reduced NADs, and 2 reduced FADs. And in the next video, we are going to be talking about what those reduced NADs and reduced FADs will be required for.